up, Jamie.
Whoa.
Jumps right out at you, right?
So sketchy.
Actually, no, it's kind of blurry right now.
It should straighten itself out. Just give it. Look away for a second. Look back. That's good. All right.
I got worse.
Yeah.
Oh, it did get better.
Ow.
All right. So this is a strand of DNA. So slice it on the hydrogen bonds.
Excuse me.
It looks like.
So the hydrogen bonds are in the middle. Hydrogen bonds are kind of weak. Why would it be good to have weak bonds here in between the nitrogen bases? Louder, Carissa.
So they can disconnect easily and turn into iron.
Yeah, so we can unzip it pretty easily to make copies of it, number one. Or number two, to read it and create a copy of RNA. Okay. That's simulation for DNA structure. Let's look at the chromosomes and the DNA.
Let's check like this.
Chromosomes, genes, and DNA. Objective. In this topic, we shall learn about the structure of chromosomes, genes, and DNA. In our body, there are many eukaryotic cells. These cells are in a state of continuous division.
Eukaryotic because they have a what?
Nucleus.
Nucleus. Good.
Now when we move inside a cell, which is about to divide, we see a conspicuous nucleus. The nucleus shows an outer nuclear membrane. As we zoom inside the nucleus, we find a nucleolus, the many thread-like chromatin. As the cell proceeds into cell division, the chromatin fibers become short and thick and are known as chromatids. These chromatins are found in pairs. The chromatids are found to be attached by a spherical structure called a centromere. The centromere, along with the chromatids, is known as the chromosome. Let us study about the different types of chromosomes. If this centromere is exactly at the center of the chromosome, such chromosomes are termed as metacentric. When the centromere is not exactly at the center, an unequal division of the arms of the chromatid occurs, and such chromosomes are called submetacentric. If the centromere is at the tail end, such a position of the chromatids is said to be telocentric. When the centromere is at the apical end of the chromosome, such chromosomes are called acrocentric. These chromosomes show a globular structure attached at the tip with the help of a nuclear organization. Such globular structures are the satellites. Seen on the chromosomes are many small fragments of genes. The position at which these genes are located on the chromosomes is called the locus. These genes are composed of large macromolecules, or the genetic material called deoxyribonucleic acid, or the DNA. Watson and Crick, the two biologists, explain the structure of a DNA. A look at the structure reveals two strands which are coiled around each other to form a double helical structure. Out of these two strands, one strand runs from 5' to 3', and the other runs from 3' to 5'.
That just means that one strand is upside down. Remember the model?
DNA is composed of
How one side
nucleotides
was
which
upside
consist of
down
nitrogen
versus
bases.
the other one?
So if you flip it over,
deoxyribose sugar,
like match
and
up
phosphate
colors,
molecules.
Not quite. The nitrogen bases would still be weird.
The nitrogen bases are purines and pyrimidines. The purines consist of adenine and guanine, while pyrimidines are composed of thymine and cytosine. Guanine always pairs with cytosine by 3-weak hydrogen bonds,
G pairs with
while adenine always pairs with thymine by 2-weak hydrogen
A
bonds.
pairs with T.
Thus, the ratio between purines and pyrimidines is always 1:1.
All right. Help us understand DNA a little bit better?
Yeah, I got a question.
Yeah, you have a
It
question?
said the nucleus. It said the nucleolus. Is that like control the nucleus?
Nucleolus is where ribosomes are made.
I know, but it like controls.
Not really. It's just where ribosomes are made. And then the ribosomes leave the nucleus and go out to the cytoplasm where they're making proteins.
Please stop.
Okay?
Stop.
Everybody good? Yeah.